Hello, I am Dr. T. Shankar, Assistant Professor of Botany, SRR, GOUTS, Government Arts and Science College, Karimnagar. Today my topic is enzymes introduction and properties. Enzymes can be defined as the biocatalysts which regulate the biochemical reactions in the living cells or organisms. The biocatalysts which regulates the biochemical reactions in the living cells or organisms. You know that enzymes are very important to the cells or living cells or organisms. They are vital for life and wide range of important functions in the cell. Without enzymes, life is unimaginable. So these are described as biochemical middleman. Enzymes also known as biochemical middleman. The term enzyme was coined by W. Kuhne in 1878. The enzyme word is a Greek word which means enzymus. En means in, zymo means yeast. The study of enzyme is known as enzymology. The study of enzyme is known as enzymology. Who studies about enzyme? Who studies about enzymes are called as enzymologists. Enzymologists studies about the enzymes. As already explained, enzymes regulate the speed and specific reactions without being used up. Because they are catalysts, they only accelerate the speed of reactions. And many enzymes, approximately 300 enzymes are made in only one cell. So living cell is having different types of enzymes. Enzymes in normal pressures, temperature and pH, hydrogen ion concentration react fastly with substrate and converts into end products. So within this enzyme plus substrate gives us to enzyme substrate complex, and now then products and enzymes are released. So this here enzymes or enzyme which is released after reaction is not affected. Now nature of enzymes. Nature of enzymes. Enzymes are chemically proteins, physically colloids. They are chemically proteins and physically colloids. Properties of enzymes. Enzymes exhibit so many properties that catalytic property, specificity, proteinaceous nature, heat stability, hydrophilic colloids, reversibility, higher molecular weight, active sites, turnover number, sensitive to hydrogen ion concentration, that is pH, inhibition of enzyme activity, and active in minute quantity. Now coming to the first property, the catalytic property, enzymes are organic catalysts. In standard environmental conditions, they accelerate the speed of the reaction. Here the enzyme reacts with a substrate and forms enzyme substrate complex, then turn into products. Second property, specificity. Enzymes are specific in their reactions. That is, one enzyme reacts with only one type of substrate but not other substrates. They do not act on other type of substrate. For example, sucrase, sucrase is an enzyme always catalyze the reaction on sucrose only. When coming to the lipase, lipase is one of the enzymes. It acts on lipids only. So here sucrase does not react on lipase and lipids. It always reacts on sucrose only. So this is the specificity of the enzyme. Now coming to the proteinaceous nature, all the enzymes are proteins, but all the proteins are not enzymes. You listen here, all the enzymes are proteins, but all the proteins are not enzymes. Those proteins which have catalytic properties are known as enzymes. The proteins which have catalytic property are known as enzymes. Based on the composition of the enzymes, there are of two types: simple enzymes and conjugated enzymes. Simple enzymes and conjugated enzymes. Simple enzymes, that means enzymes which made up of only proteins, are known as simple enzymes. Enzymes which are made up of only proteins are known as simple enzymes. For example, examples are proteases, trypsin, and pepsin. These are the simple enzymes. And second type, that is conjugated enzymes. Conjugated enzymes are made up of protein part and non-protein part. The enzymes which are made up of protein part and non-protein part are known as conjugated enzymes. So in the conjugated enzymes, protein part is known as apoenzymes and non-protein part is known as cofactor. So apoenzymes and cofactors. Apoenzyme is the only protein part in the conjugated enzymes and cofactors are non-protein parts in enzymes. Now, cofactors. Apoenzymes and cofactors together known as a holoenzyme. Apoenzyme and cofactors are together known as holoenzyme. Holo means a complete enzyme. These cofactors are two types. Cofactors two types: metal ion cofactors and organic cofactors. Metal ion cofactors, which are made up of inorganic ions. Inorganic metal ion cofactors are made up of inorganic ions like copper, ferrous, zinc, magnesium, manganese, and molybdenum. These are the metal ions. These are inorganic substances, inorganic elements. So the metal ions, which are made up of only inorganic ions, are known as metal ions. And organic cofactors. Organic cofactors are two types: coenzymes and prosthetic group. Coenzymes and prosthetic group. Coenzymes are organic factors which loosely attach to the protein part and which can be easily dialysis. Which can be easily dialysis with the coenzymes or not bound with the proteins or in inactive stage. The coenzymes are not bound with the protein part, they are in inactive stage. So if they attach to the protein part, they are active, otherwise they are in inactive stage. These are coenzymes are derivatives of vitamins or vitamin derivatives. Coenzymes are vitamin derivatives. Some examples of coenzymes are NAD, nicotinamide adenine dinucleotide, NADP, nicotinamide adenine dinucleotide phosphate, FMN, flavin mononucleotide, FAD, flavin adenine dinucleotide, PP, betaxyl phosphate, TPP, thiamine, pyrophosphate. These are the coenzymes. They are vitamin derivatives, and they and their functions are the only they help in the group transfers. Group transfers. Now coming to the prosthetic group, that is the non-protein part of the cofactors. Organic prosthetic group, which is tightly bound with the protein part, which cannot be dialysis. So heme is one part of peroxidase enzyme. This cannot be extracted through dialysis with the dialysis process. Okay, coming to the next property, heat unstability. Heat unstability. Enzymes are heat sensitive. Enzymes are heat sensitive. At low temperature, they are temporarily inactive. At high temperature, they get denatured due to their chemically proteins. The optimum temperature of enzyme activity is in a range of 25 to 30 degree centigrade. Between this temperature, the activity of enzyme is more and more. Now come next property, that is hydrophilic colloidal molecules. Hydrophilic colloidal molecules. There are two, hydrophilic and hydrophobic. Hydrophilic means attraction of water molecule attract to nature. Hydrophobic means water molecule repellent nature. So hydrophilic colloidal molecules. Enzymes are water soluble molecules. Enzymes are water soluble molecules. Water enhances the activity of enzymes and provide large surface for the reactions. Now coming to the next property, reversibility, reversible reaction. Most of enzymes exhibit reversible reactions. Reversible reactions. They form end products from substrate and vice versa. They form end products from substrate to they form end products from substrate and vice versa. So substrate to end products to substrate. Example, proteins. The presence of protein enzyme forms amino acids. The same enzyme, the presence of the amino acids are converted into proteins. Proteins are substrate, then amino acids are products. Then these are converted substrate to products. And coming to the next other example, fructose 1,6 phosphate in the presence of aldolase, they cleaved into G3P, glycerol 3 phosphate, and dihydroxy S1 phosphate. So in the presence of same enzyme, G3P and DHAP condenses to form fructose 1,6 bisphosphate. So these are the reversibility reactions and double reactions, forward reactions and backward reactions. Both reactions that takes place with these enzymes. Now, higher molecular weight due to the protein natures. Already you know that enzymes are made up of proteins. So due to this protein nature, enzymes are very large in size. They possess high molecular weight ranging from 12,000 to million daltons. 12,000 to million daltons. The daltons are the unit of the molecular weight of the enzymes. Example, catalase molecular weight is 2,50,000 daltons and urease molecular weight is 4,80,000 daltons. So every enzyme possesses their own molecular weight. Now coming to active sites or reaction centers. Active sites are also known as reaction centers. That is given as the region on the surface of the enzyme molecules which combine with the substrate is known as active sites. The region on the surface of the enzyme molecules which combine with the substrate is known as active sites or reaction centers. The shape of the active sites of enzyme will change according to the shape of the substrate molecule. Substrate molecules. So the active sites may be differ from enzyme to enzyme. Some enzymes possess one active site. Some other enzymes possess uh, more than one site, one active sites in enzyme surface. Now coming to our turnover number. The number of substrate molecules are converted into end products with one molecule of enzyme in 1 minute is called as turnover number or Tn. The number of substrate molecules are converted into end products with one molecule of enzyme in 1 minute is called a Tn number. It may be differ from one enzyme to another enzyme. Generally, it ranges from 1 to 10 to the power of 14 Tn. The enzyme Tn number is 1 to 10 to the power of 14. Sucrase has TO number, TON, turnover number is 10 to the power of 5, where hexokinase has 10 to the power of 11 and catalase 10 to the power of 6. This is the turnover number. Now come to next property, sensitive to pH. The pH, the hydrogen ion concentration. The pH concentration controls the enzyme activity to a great extent. It is dependent upon the dissociation state and charge of the enzyme. Most of enzymes work at neutral pH, that is 7. But some enzymes work in acidic, that is below 7, and some others alkaline media, that is above 7. For example, pepsin works at pH 2, peroxidase 5, amylase, urease, catalase, etc. at pH 7, and trypsin works at pH 8. So, a particular enzyme has property to work actively at certain pH numbers. Okay. Next, coming to the inhibition of enzyme activity. Enzyme activity can be inhibited by some compounds. It may be heavy metals or another compounds. Heavy metals like mercury, silver, arsenic, or inactivate the enzyme activity. Some cyanides, sulfides, and azides also inhibit the activity of enzymes. Because they break the reactions. That's why the chemical balance of chemical reaction will be deviated. Okay, last property, that is active in minute quantity. The enzymes are very active in extremely small quantities. Even enzyme size, that is a number, is very less, even though they can convert the large number of substrate molecules into products. So here, activity in minute quantity. The enzymes are very active in extremely small quantities. They convert a large number of substrate molecules into products. Okay, thank you.